We're Team 23, and for our video we focused on SKP2 expression in cancer. SCF ubiquitin ligases are E3 ligases that contain F-box proteins, such as SKP2. F-box proteins act as a substrate recognition subunit by targeting phosphorylated residues, and in combination with E1 and E2 can add ubiquitin to targets, thus playing an important role in the cell cycle. SCF-SKP2 promotes entry into S phase by regulating ubiquitination of several cell cycle control proteins, such as P27, a CDK inhibitor and tumor suppressor gene. SKP2 is a well-studied oncogenic F-box protein. Its activity is highest at S and G2 phases and is necessary for cell cycle progression. An article by Zhang et al. Investigated the role of SKP2 and SKP2B, an isoform of SKP2, in breast cancer cell growth, apoptosis, and cell cycle arrest. Figure 1 shows that SKP2 is upregulated in breast cancer samples, though it varies across different subtypes. In Figure 2, they performed a meta-analysis using Kaplan-Meier plotter outline breast cancer survival analysis. They found that high SKP2 expression positively correlates with reduced overall survival, distant metastasis survival, relapse-free survival, and post-progression survival, showing that SKP2 overexpression is associated with poor prognosis of breast cancer. In Figure 3, they measured cell viability using an MTT assay over time and detected apoptosis using flow cytometry after inserting plasmids containing SKP2 and SKP2B into cell lines. They found that SKP2 and SKP2B promote breast cancer cell proliferation and viability, in addition to inhibiting apoptosis. SKP2 activity should be high in S phase, but overexpression in G0 and G1 phase could lead to disruption in cell proliferation. In Figure 4, they used flow cytometry analysis to find that overexpression of SKP2 and SKP2B lead to cell cycle arrest in S phase and uncontrolled progression of the cell cycle. SKP2 and SKP2B are shown to regulate different downstream genes, including P27 and P53. Protein gel blotting in Figure 5 was used to determine protein levels of P27 and P53 and other proteins. SKP2 was found to seriously suppress levels of P27. Low levels of P27 is associated with breast cancer. SKP2 and SKP2B also affected the expression of P53 inducible gene 3, which is a downstream target of P53, by decreasing P53 and PIG3 levels. Low levels of these can lead to uncontrolled cell cycle progression. SKP2 did this by suppressing P53 and inhibiting PIG3 apoptosis, while SKP2B attenuated the function of PIG3 by inhibiting prohibidin, a substrate of SKP2B. Our question now is, can SPK2 be targeted to combat cancer? SKP2 affects downstream genes such as P27 and P53, leading to uncontrolled cell cycle progression and inhibiting apoptosis. Since overexpression of SKP2 is associated with cancer, it may be possible to pharmacologically inactivate SKP2 using an inhibitor to suppress its E3 ligase activity, but not the activity of other SCF complexes, in order to restrict cancer stem cells and cancer progression. Targeting DUBs could also be another potential method to combat cancer associated with overexpression of SKP2.